Hello and welcome to the presentation on PPR. PPR is a very important disease of small ruminants. It is a OI listed disease. PPR is an acronym for Pestidipeptides ruminant. It is a highly contagious viral disease of goat and sheep, characterized by fever, erosive stomatitis, conjunctivitis, gastroenteritis, and pneumonia. It has been known by many names like ovine rinderpest, goat plague, plague of small ruminants, or kata. The name PPR is commonly used worldwide, which is French word, and this means disastrous disease of small ruminants. The presence of PPR can have a serious impact on the economics of the farmer. Economic losses are due to loss of production, deaths, and abortions in infected animals. The presence of disease can limit trade, export, and import of new breeds as well as the development of intensive livestock production. In some extent, PPR has similarities with rinderpest disease in cattle and frequently manifests as fever, diarrhea, sloughing of oral and nasal mucosal epithelium, oculonasal discharge, leukopenia, and pneumonia. In natural infections, the virus causes disease in goats and sheep. However, cattle or swine remain unaffected. Goats are considered to be more susceptible as compared to sheep, but this is not always true. It is caused by morbillivirus of the Paramyxoviridae family. Morbilliviruses are important pathogens of humans as well as animals. In addition to PPR, classic members of this genus causes rinderpest in cattle, canine distemper in dogs, and measles in humans. All these are very important diseases in that particular species. Hemagglutinin and neuraminidase protein of the PPR virus helps in its attachment to the host cell receptor, and it is found that it has very high affinity towards the SLAM as well as nectin-4 host cell receptor. SLAM, that is signaling lymphocyte activating molecule receptors, they are generally present on antigen presenting cells, and nectin-4 is a host cell receptor which are generally present on the epithelial cells. That's why this virus has got affinity toward both lymphoid as well as epithelial cells. In this disease, morbidity varies from 80 to 90% and mortality may go up to 80%. However, all the animals in a particular flock may produce disease and if live untreated succumb to death. More severe disease is seen in young animals as well as animals with poor nutrition and concurrent parasitic or bacterial infections. PPR is an economically significant disease and great threat to the goat industry in the country. It was first reported in the Ivory Coast of Africa in 1942, and since then it has just spread to various countries from Africa, Asia, and Middle East. It is one of the important diseases of small ruminant in India also. Generally, transmission of PPR virus occurs by inhalation of contaminated aerosols from closely associated animals, and it is thought to be an important means of disease transmission. Moreover, it can also spread by ingestion of contaminated food and water or by direct contact through licking and nuzzling, and occasionally through fomites such as water trays, feed bunks, which are recently used by infected animals. Then how this virus produces disease in that particular susceptible host? Virus enters through inhalation or ingestion. After entry of this PPR virus, the virus is initially taken up by these antigen presenting cells which carry SLAM receptor present in the intraepithelial space and lamina propria of respiratory mucosa, that is nasopharyngeal mucosa and respiratory epithelium, from where it is transported to the regional lymphoid tissues where primary replication takes place. Then via infected lymphocytes, infection spreads throughout the body via lymphatic and vascular system. PPR virus is both lympho as well as epitheliotropic, and infection usually result in conjunctivitis, rhinotracheitis, ulcerative stomatitis, gastroenteritis, and pneumonia. Like other morbilliviruses, PPR virus is also cell associated. Therefore, it is thought that it reaches to other visceral organs and tissues through peripheral blood mononuclear cells. The lung infection with PPR virus is considered as late event occurring in the face of high viral load. PPR virus lead to extensive necrosis in lymphoid organs, that is Bair's patches, spleen, thymus, and pulmonary lymph nodes, and hence results in reduction in circulatory peripheral blood leukocytes, that is also called as leukemia. Virus has also been found to induce apoptosis in peripheral blood mononuclear cells, and this is how this virus produces various ulcerative erosive lesions in different organs as well as damages the lymphoid organs and lead to the immunosuppression. Regarding clinical signs, incubation period of PPR virus varies from 2 to 10 days. However, 2 to 6 days considered to be typical. It can be exhibited in various clinical forms, and this is decided by the strain of that infecting virus, immune status of animal, and variable stressors, environmental stressors or managemental stresses. For acute cases can be seen when PPR first occurs in naive populations. Those animals which have never been exposed to PPR virus, such population of animal is called as naive population and characterized by sudden death. Most cases of PPR are acute in nature. The characteristic signs are high fever with serious nasal and ocular discharge that may become mucopurulent over the time. Matting is common around the eyes and nose may become obstructed. Within a few days of the onset of fever, the gum become hyperemic and small grey necrotic foci covering shallow erosions begins to appear in the mouth. Most animals develop profuse diarrhea which may be watery or blood-stained with very bad odor and sometimes contain shreds of slough tissues. Signs of pneumonia like rapid respiration, dyspnea and coughing are seen commonly. Some animals may abort. In the late stage of disease, small nodules resembling contagious eczema, sheeppox or goatpox can appear in the skin around muzzle. Severely affected animals become dehydrated and emaciated. Hypothermia can precede death. Animals that do not die often have prolonged convulsions. In subacute disease, immunosuppression led to secondary bacterial infections and which may aggravate the condition. This form usually lasts for 10 to 15 days. The signs are variable but often include respiratory signs. Asymptomatic infections can also occur in these animals. Regarding gross and microscopic lesions produced by this PPR virus in the infected host, pathological changes associated with PPR virus includes erosive stomatitis and enterocolitis, similar to that of rinderpest in cattle. That's why it is also called as ovine rinderpest. And proliferative and necrotizing bronchointerstitial pneumonia. Lymphoid depletion or necrosis occurs in the spleen, bare patches and variable lymph nodes. In fatal cases of the disease, pneumonia may not be as severe in young goats than in older animals, probably because young kids succumb to dehydration caused by diarrhea before pulmonary lesions can fully develop. PPR-induced pneumonia shares several histologic features with the lung lesions of distempers in dog and measles in human. Bronchointerstitial pneumonia occurs and characteristic large multinucleated cells, that is also called as syncytial cells, containing intracytoplasmic and intranuclear inclusions are found within the alveoli of the lung as well as in the terminal bronchioles. As virus is responsible for the destruction of lymphoid organ, it causes immunosuppression and subsequently secondary bacterial infections which may worsen the condition. To just diagnose this disease, you have to collect the various tissue discharges, excretion or secretions from the infected animals as well as oral lesion discharges from the oral lesions, and whole blood can also be collected. To diagnose this disease, clinical signs, gross and microscopic lesions helps, but to confirm the disease we have to rely on the laboratory test which includes antigen detecting test and antibody specific to this virus detecting test. It includes reverse transcriptase PCR, real-time PCR, ELISA, agar-gel immunodeficient test, virus isolation. So by using these all laboratory techniques, we can just confirm the disease as PPR. Thank you.